Hello students, let's discuss about solid phase peptide synthesis. Introduction. The synthesis of peptides in the laboratory has been a major biochemical objective for many years. But while the synthesis of polypeptide hormones has been achieved, a chemical synthesis of a natural protein in vitro is an unconquered Everest. From the practical point of view, such synthesis might well provide variants of polypeptide hormones and proteins with great medical potentialities. Nowadays, peptide synthesis includes a large range of techniques and procedures that enables the preparation of small peptide to large peptides. The pioneering work of Bruce Merrifield, which introduced solid phase peptide synthesis, also known as SPPS, dramatically changed the strategy of peptide synthesis. After defining a synthesis of strategy and programming, the amino acid sequence of peptides machines can automatically perform all the synthetic steps required to prepare multiple peptide samples. SPPS has now become the method of choice to produce peptides. Let us discuss about Bruce Merrifield's synthesis of solid phase peptide synthesis, also known as automated peptide synthesis. In 1969, Bruce Merrifield described a method that revolutionized the synthesis of peptides because it provides a much faster way to produce peptide in much higher yields. He received the Nobel Prize in Chemistry for developing automated solid phase peptide synthesis in the year 1984. Furthermore, because it is automated, the synthesis requires fewer hours of direct attention. Using the technique, bradykinin was synthesized with a 85% yield in 27 hours. Subsequently, refinement in the technique now allow a peptide containing 100 amino acids to be synthesized in 4 days in a reasonable yield. In the Merrifield method, the C-terminal amino acid is covalently attached to a solid support containing in a column. Its N-terminal blocked amino acid is added one at a time along with other needed reagents. So the protein is synthesized from the C-terminal end to the N-terminal end. Notice that this is opposite to the way of protein are synthesized in nature, that is from the N-terminal end to the C-terminal end. Because it uses a solid support and is automated, this method of protein synthesis is called automated solid phase peptide synthesis. The solid support to which the C-terminal amino acid is attached is a polystyrene resin similar to the one used in ion exchange chromatography, except that the benzene ring have chloromethyl substituents instead of sulfonic acid substituents. Before the C-terminal amino acid is attached to the resin, its amino group is protected with tertiary butyl oxycarbonyl to prevent the amino group from reacting with the resin. The C-terminal amino acid is then attached to the resin by means of an nucleophilic substitution reaction, SN2 reaction. Each carboxyl group attacks a benzyl carbon of the resin, displacing a chloride ion. After the C-terminal amino acid is attached to the resin, the TBOC protecting group is removed. The next amino acid with its amino group protected with tertiary butyl oxycarbonyl and its carboxyl group activated with DCC, dicyclohexyl carbodiamide, is added to the solution. A huge advantage of the Merrifield method of peptide synthesis is that the growing peptide can be purified by washing the column with an appropriate solvent after each step of the procedure. The impurities are washed out of the column because they are not attached to the solid support. Because the peptide is covalently attached to the resin, none of it is lost in the purification step. leading to the high yield of purified product. After the required amino acids have been added one by one, the peptide can be removed from the resin by treatment with HF under solid conditions that do not break the peptide bond. This technique is constantly being improved so that peptide can be made more rapidly and more efficiently. However, it cannot compare with nature. A bacterial cell is able to synthesize a protein thousands of amino acids long in seconds, and it can simultaneously synthesize thousands of different proteins with no mistakes. Since the early 1980s, it has been possible to synthesize proteins by genetic engineering techniques. Strands of DNA can be introduced into bacterial cells, which causes the cells to produce large amount of a desired protein. For example, mass quantities of human insulin are produced from genetically modified E. coli. Genetic engineering techniques also have been useful in synthesizing proteins that differ in one or a few amino acids from the natural protein. Such synthetic proteins have been used, for example, to determine how a single amino acid chain can affect the properties of a protein. Now let us discuss about the advantages of solid phase peptide synthesis. Advantages of solid phase peptide synthesis is that the product can be isolated easily since all the intermediates are immobilized on polystyrene. Thus, peptides can be purified by filtration and washing. Repetition of the deprotection coupling process will be able to synthesize larger peptides. Synthetic peptides are made for many purposes. These peptides can act as antigens, which will stimulate the immune system of the body to produce antibodies that target such peptides. These antibodies can then be used to isolate a protein. Peptides can isolate receptor hormones. Synthetic peptides can be used as drugs and to gain a greater understanding of the three-dimensional structure of proteins. In solid phase synthesis, the peptides have now been discovered to have important roles in biology. Now let us look into more about applications of solid phase peptide synthesis. Peptides are synthesized for a variety of applications, areas, and research interests. Peptide synthesis application area include array of library synthesis, synthesis of peptide for biological or screening arrays, and screening of reaction conditions. Solid phase peptide synthesis is a favorite route for peptide synthesis. SPPS is used for interesting applications like synthesis of cyclic peptides, polypeptide libraries, synthesis of libraries of protease substrates, screening of reaction conditions like coupling time 3 seconds, concentration of reagents, neutralization of optimization of high value synthetic processes are possible. Some applications such as drug development, industrial synthesis of peptides, therapeutic peptides, selective robotic assisted modifications of NPY receptor ligands are as follows. Application of automated SPPS for drug development. The described method of chemical synthesis of peptide on a solid phase, and particularly its outstanding potential for automation, have led to routine method in the development of novel pharmaceuticals. In principle, there are two approaches for drug design: rational and combinatorial methods. While the rational procedure is a lead structure-oriented process, there is typically little knowledge about the evaluated biological system in combinatorial methods. In order to identify a lead compound of a relative unknown system, numerous molecules also known as peptides, have to be produced in parallel by a combination of building blocks creating a peptide library. The first parallel synthesis of hundreds of peptides was published by Jason et al. In 1984. Peptide libraries can be created by directed parallel synthesis of complex peptide mixtures and identified by position screening. In the industrial synthesis of peptides and alternative production methods, the successful automation of peptide synthesis led to a breakthrough on the peptide therapeutic market and vice versa. The great effort in improving linkers, protecting groups, resins, etc. provided access for the synthesis of larger peptides and even proteins. But nevertheless, the sequential and convergent production of therapeutic application is often the only possibility for manufacturing peptides which are larger than 50 amino acids. This method is based on the independent synthesis of fully protected linear peptide fragments being selectively condensed in solution to obtain the desired polypeptide. The condensation can occur via chemoselective ligation technique such as native chemical ligation, also referred to as NCL, expressed protein ligation referred to as EPL. Recently, peptide drugs as the pharmaceuticals enfuviritide, eptifibatide and bivalirudin have been manufactured in multi-kilogram scale. Enfuviritide is an efficient membrane fusion inhibitor of HIV treatment consisting of 36 amino acids. Salmon calcitonin, human glucagon and human insulin are polypeptide being commercially produced by recombinant expression. In general, the quality of chemically synthesized peptide therapeutics is comparable to recombinantly or enzymatically produced compounds. Let us discuss about the next topic, which is the combination of automatic and manual SPPS to obtain therapeutic peptides. The majority of peptides are hormones being responsible for a broad scope of physiological functions. Here, we highlight two successful strategies to modify chemical properties in order to influence pharmacodynamics and kinetic profiles: lipidation and PEGYLATION. As an example, modern concept of SPPS, assisted selective derivatives, is described for neuropeptides Y, also referred to as NPY, receptor ligand for therapeutic and analytical applications. In modification of therapeutic peptides to extend their half-lives, peptide stability can also be optimized by blocking their receptive termini through N-terminal acylation and C-terminal amidation. Apart from this, metabolically unstable peptide drugs can be optimized by the covalent attachment of fatty acids, also known as lipidation, or methoxypolyethylene glycol or PEG polymers. These two strategies are based on substantially different mechanisms which can lead to a remarkable increase of the potential utility of peptides as pharmaceuticals. Lipidation of peptides. In general, the half-life extension of peptides by lipidation is obtained by an increased binding to albumin, which is the most abundant protein within the extracellular fluid. Human serum albumin is a fundamental carrier of non-esterified free fatty acids as well as multiple other endogenous ligands and drugs in the blood. Here, insulin was acylated with saturated fatty acids containing 10 to 12 carbon atoms at the beta chain using the epsilon group of the lysine. In this study, an increased albumin affinity of lipidated insulin variants depending on the number of carbon atoms by interaction studies with immobilized human serum albumin. Moreover, they were able to show a sustained lowering effect of blood glucose, demonstrating a prolonged action profile of the acylated conjugates. Since then, many biological relevant peptides and proteins were chemically modified by fatty acid acylation. PEGylation of peptides. In another modification of the peptides, PEGylation is the covalent modification of peptides with methoxy polyethylene glycol polymers. PEGylation is the covalent modification of peptides with methoxy polyethylene glycol polymer units. PEG itself is known to be amphiphilic, non-toxic, little immunogenic, non-antigenic, and highly soluble. These beneficial attributes are conveyed to the peptide by covalent attachment, leading to an increased size which lowers renal ultrafiltration. Moreover, the PEGylated peptide is surrounded by a large water cloud owing to the capability of every ethylene oxide unit to bind about 3 water molecules. These lead to an improved solubility and to a remarkable shielding of the peptide against protease and antibodies that is reflected in an improved stability and biodistribution as well as reduced immunogenicity. A conventional strategy for peptide PEGylation is the use of the SPPS technique. Here, polypeptide with specifically protected side chains are synthesized by automated SPPS according to the strategy that is applied for the introduction of fatty acids. It has been shown that the amino acid position plays a pivotal role for maintaining the biological function. The incorporation of a 40 kilodalton branched PEG to interferon alpha 2A has led to one of the first launched PEGylated drugs, which is used as an antiviral drug for the treatment of hepatitis C nowadays. In the next topic, we have selective robot-assisted modification of NPY receptor ligands. The 36 amino acid peptide hormone neuropeptide Y, NPY, peptide YY, and pancreatic polypeptide PP are endogenous ligands of the so-called NPY family. They are responsible for a variety of physiological functions such as food intake, energy homeostasis, cancer, cell proliferation, blood pressure, and epilepsy. Those effects are mediated by 4 distinct G protein coupled receptors Y1, Y2, Y4, and Y5 that are expressed in central and or peripheral tissues. In conclusion, the principle and the different steps of SPPS are all discussed along with their possible side reactions. Solid phase peptide synthesis, SPPS, is a favorable route for peptide synthesis. The peptides have now been discovered to have important roles in biology. The production of peptides by automated synthesis on solid support provides a great variety of benefits. Chemical reactions necessary to assemble peptides can be performed simultaneously with plenty of reaction tubes, allowing parallel and multiple synthesis. Owing to the simple washing of the resin following coupling or deprotection steps, no intermediate purification is necessary. Automated SPPS is of great importance, especially for structure activity, relationship studies, and backbone modifications of biologically active peptide hormones. Although remarkable progress has been accomplished, Since the invention by Merrifield more than 50 years ago, novel technologies in automated peptide synthesis are required. This technique is progressing but needs more improvement, especially in terms of practicability. The ongoing need for peptide as biopharmaceuticals will surely promote this development in the future.